let's discuss the shared common structural features of the amino acids. In this part, we will be discussing that structure of the amino acids which is shared by almost all the types of the amino acids which have been discovered up till now. There are two exceptions of the amino acids, such as proline and glycine, which show deviation from this rule because they have a structure that is quite different from this general structure. And when we will discuss their structure, we will discuss this reason soon. So first of all, in this part, if we see the general structure of the amino acid, there is a central carbon which is called as alpha carbon, and this central carbon is also called as chiral carbon. Why we call it as a chiral carbon? A chiral carbon is the carbon that can attach to the four different groups at one time. So as it is shown in the diagram, that this central carbon or the alpha carbon is attached to the four different groups, and you know that all these groups are completely different from each other. That's why this central carbon is also called as chiral carbon, or the center region is also called as a chiral center of the amino acid. And this chiral center is very important to determine the chirality of the amino acids, and in the next lecture we will discuss the chirality of amino acids. This part would have the prime importance. So this is the first part of the amino acids. Now let's discuss the four groups which are attached to this chiral center one by one. So first of all, we have this carboxylic group which is attached to this chiral center. And as you know that carbon has a valency of 4, so definitely it can make the 4 covalent bonds with all these 4 different groups. So next, there is another important functional group that is called as amine group that is present here. So this is the carboxylic group. And then there is a hydrogen atom that is available here. This will make the third group. And last, one of the most important group is called as R chain. It is also called as aromatic chain. You can also say it as a hydrocarbon chain, alkane chain. So it can exist in a variety of the forms. It can exist in the ringed form, it can exist in the double bonded form, triple bonded, and single bonded forms as well. So in general, you can say it's a hydrocarbon chain, and for simplicity, we just named it as R. What is the importance of this R chain? Why it's so important for the amino acids? Because it is actually the R chain that determines the type of the amino acid. So R chain is actually responsible for determining the type of amino acid. And why it determines the type of the amino acid? Because in each amino acid, this R chain is different. So every amino acid that you will study, when you will study the structural features, each of that amino acid will have this R chain basically different, and that is very important. So up till now, almost more than 500 amino acids which have been discovered, all of these 500 amino acids will have this different R chain or R group, and that's why it would be different from the other amino acids. So that is very important to consider. Now, the structure which I have told you here, or we have discussed, this is the unionized or unpolarized or uncharged structure of the amino acid, and it is just a type of the structure which we usually discuss when we are studying the structural features of the amino acids. But this structure is least common structure. This type of the structure usually doesn't exist, or you can say the amino acid doesn't exist in this particular form, because I have already told you that amino acids are usually involved in making the biological molecules, and biological molecules are usually existing in the bodies of the living organisms. And in the bodies of the living organisms, you know that there is existence of the certain pH values. For example, if we talk about the human body, then within the body of the human beings, there is a buffer system, you know that, and the physiological pH of the human body at which all the enzymes and metabolic reactions can take place, that physiological pH is 7.4. So at this 7.4, what is actually the condition, or what is actually the appearance of the amino acid? Um, I'm going to come to this structure later on. First, I'm going to make it clear that how this structure is basically going to convert into a different type of the structure at a particular pH of the 7.4, and what would be the changes in that structure when the pH actually changes. So let's discuss its structure at the pH of 7.4, that is the human body's pH. Okay, so first of all, very important thing, at the pH of 7.4, you know that the concentration of hydrogen ions is equal to the concentration of the hydroxyl ions, and that is from the basic organic chemistry. Now let's draw the structure of the amino acid again. So there is a C alpha carbon, which I have already drawn, and next we have these four different functional groups to which an amino acid is basically attached, and there is a carboxylic acid group, there is an R group, there is a hydrogen atom, and then there is a amine group that is present. Okay, now at this particular pH, this carboxylic acid, as you basically can easily suggest from its name, that it will exist as an acid. That is a very important property. Now, what is the property of an acid? Acid has the ability to lose hydrogen ions, or you can say it has the ability to lose the protons. Okay, so there will be the loss of hydrogen ions from this carboxylic group, and when the hydrogen ion is lost, when this hydrogen ion is lost, mean one proton has been lost, so it will doesn't exist in this particular stage, rather the carboxylic acid will exist C double bond O, and with one extra negative charge on it. Okay, so at the physiological pH of the 7.4, when the hydrogen ions and hydroxyl ions concentration in the system is equivalent and these ions are available in the system, then carboxylic acid will have the ability to lose its hydrogen ions in the medium, and it will behave as an acid. Now let's move towards the second part. Now here we will have a amine group. An amine group at this physiological pH will behave as a base. And what is the property of the base? Base has the ability to accept protons, that is the hydrogen ions. So it has the ability to accept the protons, and when it will accept one proton, mean it will accept one hydrogen ion. Okay, when it will accept one hydrogen ion, so it will no longer exist in this particular state, and NH2 will be converted to NH3, and as one extra positive charge or one proton has been added to its structure, so it will have one extra positive charge on its molecular ion. Okay, so now this is actually the state at which now amino acid is no longer in the unionized form. Now the amino acid has become, you can easily say that now the amino acid has become ionized. And this state of the amino acid is actually called as Zwitterion state. So you can say at the physiological pH in the human body, the amino acids actually exist in the Zwitterion state, or it is a dipole state. Why it is a dipole state? Because in this amino acid, there is one negative pole is created and one positive pole is created. And as the quantity of the charge, that is the negative charge and positive charge, is almost equal, is equivalent to each other, so the net charge on amino acid would be 0. Still, it would be a dipole species, or dipole state, in the dipole state, because two poles has been created. One is the negative pole and the other one is the positive pole that is basically created. But the overall charge on the Zwitterion or on the amino acid would be considered as 0. Now, for example, we place this Zwitterion. For example, this is an amino acid, and we have charges on it. We have a positive charge on its one pole and we have a negative charge on the other pole. And we place this Zwitterion in a electric field. And in the electric field, you know that there are two poles. In the electric field, there are basically two poles. So there is one positive pole is there, or positive electrode is there, and the other one is called as negative electrode is there. So what you say that this amino acid which has a dipole state, how it is going to move, in which particular direction it is going to move, whether in this direction towards the negative pole state or towards the positive pole state. So the answer is that the amino acid is not going to move in any of these directions. Rather, it will remain as still. It doesn't show any movement. So at that point of pH, where in an electric field the amino acid doesn't show any movement and the net charge on it is 0, that particular pH of the amino acid is called as its isoelectric point. So how would you define the isoelectric point? Isoelectric point, that pH of an amino acid on which net charge on that amino acid is actually 0, and it doesn't show any movement, or it doesn't move towards any pole of the electric field. So that is basically the definition of isoelectric point. So let's move towards the structure which I have taken from the book Lehninger Principles of Biochemistry. So now you can basically understand this, how we basically move from this particular structure. I have told you from this particular structure how we have moved to this particular structure. So this is basically the ionized form of the amino acids. So in the human, at the physiological pH of the 7.4, the amino acids basically exist in this state. It has a central alpha carbon. It has basically a carboxylate ion group. Then there is a hydrogen ion. Then there is basically the ammonia ion, and there is an R chain. So basic difference which basically occurs and in the groups, that is the amine groups and the carboxylic acid group, because both were ionizable. Carboxylic acid show the acidic nature and it gets converted into the carboxylate ion, and similarly the amine basically shows the base group, which accept a base property. It accepted actually the one proton and it is now a ammonium ion. So that is how basically the amino acids exist at the physiological pH. Now let's talk about the two other scenarios. I have already discussed here that this particular state in which the pH of the amino acid is like that, in which it doesn't show any movement in the electric field, that pH of the amino acid is called as isoelectric point. But it means that there is not any proper fixed pH for each amino acid at which it doesn't show any movement. So up to now we have discussed here that at the physiological pH, the overall net charge on the amino acid will be considered at 0. And when that amino acid is placed in an electric field, it doesn't show any movement. And that pH at which amino acid doesn't show any movement in an electric field, that is called as its isoelectric point, RPI, and that is different from for every amino acid. But here we have discussed a condition of the physiological pH, that is 7.4, that is almost considered to be the neutral for the human body. Now let's discuss the same condition of the amino acid but at the different pH values. And you know that the two other pH conditions that can happen is that we can have the acidic pH of the medium and we can have the basic pH of the medium. So for this, let's draw a pH scale here. Okay. So there is a center point that is pH of 7, that is considered to be neutral, and around this almost 7.3 and 7.4 is the physiological pH of the human body. And above that, there are values like 8, we have 9, 10 pH, 11, 12, 13, 14, and similarly the low pH values such as 1, 2, 3, 4, 5, and 6. So this is the overall pH scale which we are considering right now. Okay, so at the neutral pH we have already discussed a case that what is going to happen. The overall charge in the amino acid would be 0, and that would be its isoelectric point. But what happens if we move towards the lower pH values? And what will be the charge of the amino acid when we move towards the higher pH values? And what is mean by this? When I'm going to talk about the pH value below 7, like if there is a pH of the medium like 6, 5, 4, 3, 2, 1, that pH is considered to be acidic pH. So if I say that I have a medium, I have this medium, and this medium has the acidic pH. This medium has actually the acidic pH that can be 1, 2, 3, 4, 5 up to 6. Whenever the medium has acidic pH, the concentration of the hydrogen ions is high. The concentration of the hydrogen ions in that medium is high, and the concentration of the hydroxyl ions is low. While in the neutral pH, you have considered that almost at the pH of 7.4, the concentration of the hydrogen ions and hydroxyl ions are almost equal. Okay, so here in the acidic medium, the scenario is different. Now, if we have an amino acid, so let's draw here. We have an amino acid, and it is attached to the 4 different groups. So one, you know that, let's suppose that consider that intermediate charge of the at which the pH at which the amino acid is present at the neutral pH, like there is a carboxylate ion, there is ammonium ion, NH3 positive, there is R, and there is H. Let's suppose that amino acid is present at the pH of the 7.4, and suddenly the pH of the medium has become acidic. And now this is the condition of the medium, and the concentration of the hydrogen ions and hydroxyl ions are not equal, and the concentration of the hydrogen ions is actually high as compared to the concentration of the hydroxyl ions, which is low. Okay, now what is going to happen with this particular amino acid? Now this amino acid will undergo a particular condition that is called as protonation. So what is going to happen here? There is the protonation of the amino acid that is going to happen. So what is protonation? Protonation is the addition of protons, mean hydrogen ions. So there will be the addition of the hydrogen ion wherever it can be added in the structure of the amino acid. So in the structure of the amino acid, you can already predict that where the hydrogen ions can be added at this particular point. So carboxylate ion will actually accept the hydrogen ions because the concentration is high and it has one extra negative charge on it. So the hydrogen ions has the ability to be accepted by the carboxylate ion in this negatively charged state, and carboxylate ion will be converted from this state to this state. So here you can say that you can actually see that this pole now has no charge, while this pole has one extra positive charge on it. So now the overall charge on the amino acid will now be plus 1. So when just you have to consider this point in your mind, that at this stage the amino acid actually behaves as what? Acid or base? It has accepted a proton, so it behaves as a base, and it will have one extra positive charge on it. So in the base, in the acidic pH values, the amino acids behaves as base, and they have one extra positive charge on them, so they exist as positively charged species or amino acids. Okay, now let's discuss the other scenario. And in this particular scenario, we have a medium, and I'm saying that my medium has basic pH. And when the medium has a basic pH, you know that the concentration of the hydroxyl ions would be high. That could be the pH of any medium of any living organism body, even of any part of the organ as well. So the concentration of the hydroxyl ions would be high, while the concentration of the hydrogen ions would be low. Okay, so now again we have an amino acid, we have alpha carbon, and it is attached to the 4 different groups. Like we have carboxylic group, we have hydrogen atom, we have R group, and we have amine group. Sorry, so let me draw here like intermediate state, and we have this carboxylate ion. Okay, so now as the concentration of the hydrogen ions are low and they are not frequently and freely available for the protonation, so here actually in the medium the deprotonation basically takes place. And in which part of the amino acid actually the deprotonation can take place, where the extra proton is actually available, in this particular region the extra proton is basically available. So this part will undergo the deprotonation, and one hydrogen ion will be lost. So it will no longer exist as ammonium ion, it will now exist as NH2. So that is basically the condition. Now the charge on this pole is now 0, so it doesn't have no longer any kind of the positive charge. There is one extra negative charge available on this pole, so overall charge on the amino acid would be negative. So in case of the basic pH values, the amino acids behave as acid, because acids has the ability to lose the protons. So it's in this particular region it has actually loss of protons, so there is loss of the protons. Okay, so it behaves as an acid and it will have overall negative charge on it, so it should be considered as a negatively charged species. So I hope this is clear to you, that at the neutral pH values, that is at pH of the 7.4, 7.2, 7.3, the amino acids exist as in the Zwitterion state, where the quantity of the positive charge and negative charge is almost equal, and the overall charge on the amino acid is 0, and it doesn't show any movement in the electric field. While in the acidic pH values, the amino acids in the acidic medium usually behave as a base. They actually accept the protons in this particular case, okay, and they behaves as an acid. While in case of the basic pH values, they actually lose the proton and the overall charge on the amino acid is negative and they behaves as an acid because they lost the protons. Now, very simple rule, if you have a electric field here and you know that there is a positive pole and there is a negative pole, and here is your amino acid, and I'm talking about this state when the amino acid has an overall positive charge on it. Okay, so what will you suggest that in which particular direction amino acid will move when the electrodes are turned on and they are charged? Definitely the amino acid is going to move towards the negative pole. Okay, and similarly let's consider another scenario, and here I'm going to consider this scenario, and the overall charge on the amino acid is negative. So now in which particular direction the amino acid is going to move? Definitely the amino acid is going to move towards the positive pole. So in case of the basic nature of the amino acids, when the amino acid has a positive charge on them, they move towards the negative pole when the electric fields are turned on. And similarly, when they behave as an acid and they have negative charge on them, they move towards the positive pole when the electric fields are turned on. So that was all about today's lecture. And in the next lecture series, we will be discussing the chirality, nomenclature, the structure of the amino acids, and many more about the proteins, peptides, and their structures. So thank you so much.